This is part two of week four, the biology lectures. We're talking about the kingdom Monera, and now we're going to talk about how these organisms make and consume energy. First of all, we have to have some definitions, and the first definition I'd like to introduce is the idea that of an organism that feeds on dead matter. We spoke in our previous lectures about producers, consumers, and decomposers. And bacteria is probably the largest group that makes up most of the decomposers in our environment. And saprophytes are those organisms that feed on dead material. And this is just one example of a saprophyte. Now this saprophyte actually is not in the kingdom Monera. This is one of the fungi, which we will talk about in a future lecture. But this plant does not have any chlorophyll, so it cannot produce any energy on its own. But it has to obtain its energy from other places, and basically it grows and it extracts its energy from the dead material that's in the ground around it. Now, a parasite is different from a saprophyte in the fact that a parasite lives on a human host. And generally, although ultimately it may cause the death of that host, in many cases it does not cause the death of the host. It does cause harm, though, and the difference between some organisms that live with other organisms and they mutually benefit from that interaction, a parasite gives no benefit to the living organism that it's feeding on. And here is an example of a parasite. This is a tick that's extracting blood from a human arm in this case, and the human achieves no benefit, and therefore that this would be considered a parasitic interaction. If somehow the human were to receive benefit, then this would not be called a parasite. We've already spoke before about this. Organisms that do not require other organisms in which to live, but achieve their energy through the change of light energy into food, do this by means of photosynthesis. And there are groups of organisms in the kingdom Monera that do photosynthesis. And the last thing I'll mention is the idea of chemosynthesis. Chemosynthesis is a different process to form food. In other words, instead of using light energy as the energy source comes from something else and is converted to food energy in one way or another. Now this slide shows the difference between anaerobic and aerobic respiration. And most organisms that undergo either anaerobic or aerobic respiration have to undergo a process known as digestion. Digestion is a process where larger food molecules that are absorbed from the environment are broken down into smaller molecules that can be used to form energy. And it's important to understand the difference between a digestion and absorption. Digestion is when these food molecules are actually broken apart, and absorption is when they're taken into the body. And respiration is when the food molecules are then converted into actual energy. Digestion is not converting them into actual energy, but it's merely the breaking down of a large food molecule into a smaller food molecule. For example, the food we eat is made up of large chains of carbohydrates, which are made up of chains of glucose and fructose. Digestion is the process of breaking those large chains down until you have just glucose and fructose, which is then absorbed into the body and converted into energy by the means that from the lecture, our last lecture. Now, an aerobic organism does these processes and requires oxygen when it does these processes. And if you look at the middle column, it talks about what makes an aerobic organism aerobic. First of all, it requires oxygen. Secondly, it produces carbon dioxide and water. Thirdly, it tends to be a very, very efficient form of energy in terms of the amount of energy per glucose used. And lastly, it tends to produce heat. The other form is anaerobic organism, and that organism does not necessarily require oxygen. Now, some are called microaerophilic, which means they need a little bit of oxygen, but others do not require and actually do not thrive in an oxygen environment. And as you can see, the anaerobic does not need or want oxygen. The side products or waste products are different in that we still get carbon dioxide, but now, as we've talked about before, we also produce ethanol and in some cases lactic acid. This is less efficient, which is why lactic acid metabolism is not preferred, because some of the energy is lost in the lactic acid or in the alcohol. And yes, heat is released, but again, less than what is seen with aerobic metabolism. The other thing that's very interesting about the kingdom Monera is how they reproduce. Now, the way they reproduce is generally very simple. It's by a process known as binary fission. What happens in binary fission is first the DNA coil is replicated, then the two cells divide into two, and you end up with two daughter cells that are identical genetically and in every other way to the original parent cell. Generally, what tends to happen is that when an organism is introduced into an environment, it grows fairly rapidly until it reaches a point where the number of organisms that are being produced is the same as the number of organisms that are dying. And this is called a steady state. This is a state where the members die at the same rate that new ones enter the population. And this generally is controlled by the amount of food or other resources that are necessary for life that's in the environment. Now, as I said, initially when you introduce an organism like a bacteria into an environment, it grows, and it grows exponentially. That is, 1 becomes 2, 2 become 4, 4 become 8, etc. And this unhindered growth is due to the fact that there's far more resources available than are necessary to support the population. And this is can be drawn graphically with a curve that gets larger and larger as the population gets bigger and bigger. But at one point, the resources become more limited, and at that point, the growth is called logistic, and it slows down. And then ultimately, you reach what I've stated at the top here, which is a steady state, where the population plateaus in terms of the number of living organisms that are present. And generally, all of this is controlled by the amount of resources that are available in the environment. Now, it's very interesting. Organisms can do several different things in terms of exchanging DNA with each other. And one means in which this is done is called a conjugation. In conjugation, two bacteria temporarily unite with each other, and there's a transfer of the DNA from one organism to the other. And you remember I told you that there was extra DNA, and here it's shown here, and that extra DNA is called a plasmid. And this is a small circular aspect of DNA that's separate from the main coil of DNA. And that's generally the DNA that's exchanged between the two organisms. And this exchange occurs through something called a conjugation tube. And the conjugation tube is the communication between the bacteria, and the DNA is transferred. And you remember I mentioned the fimbriae that was called a sex pilus, and this basically unites these two organisms together, and then there's an exchange in DNA across from one organism and another. And this was first found in a human setting when they saw the transfer of bacterial resistance from a resistant bacterial strain to a sensitive bacterial strain without the sensitive strain was not exposed to the bacteria, which is how resistant strains are generally created. Now, what I mean by that is if you want to make a organism resistant to an antibiotic, you expose repeated generations to that antibiotic until you have a group that survived the antibiotic. And however it survives is generally coded in its DNA, and it's frequently coded in the plasmid part. And what they found is that this resistance could be transferred without exposure of the recipient to the bacteria. And that's one of the ways that this was originally discovered. Other ways in which DNA can be transferred are called transformation. Now, this is when, in our previous example here, these two organisms are both functional. Okay, they're both alive and kicking and doing well. But in transformation, the DNA transfer is from a non-functional donor to a functional resistant. So you see there's a fragment of DNA that's in the environment. The cell itself has actually died, and this is somehow taken up into the donor or recipient cell, and it incorporates that DNA into its DNA. And this was actually shown. What they did, it was an experiment where they actually killed bacteria, and these were bacteria that were resistant to antibiotics, and they just put the lysate or the dead stuff from the bacteria in a medium and put live bacterial sensitive or antibiotic sensitive bacteria in there, and they showed that they could generate resistance to antibiotics by the incorporation of this DNA, so by this transformation. Now, the third way is called transduction. Now, transduction is the transfer of DNA by means of a viral infection. Viruses are very simple organisms that have an outside cell wall, and they have DNA. And they essentially attach themselves to another living organism, and they take over the machinery of that cell and force it to replicate the viral DNA, thus propagating the virus. And so here's an example. This is called a phage. A phage is a virus that infects bacteria. You have a little piece of DNA here, and it attaches itself to the bacteria and injects the DNA, and then this DNA is incorporated into the bacteria. It produces more virus, but this next infection occurs and incorporates into the bacteria. So ultimately, the bacteria become not only used to make new virus, but also incorporate this DNA into its DNA. So there are three ways in which DNA is transferred from one bacterium to another. One is conjugation, where two live bacteria transfer. The second is transformation, in which DNA is transferred from a non-functioning donor to a functional recipient. And then transduction, in which a virus is used to transfer DNA from into a bacterial cell. Now, the other thing that's kind of interesting is that bacteria can actually incorporate its DNA into a large organelle of sorts that's inside its body, and then surround it with a hard core. And then this core and this becomes known as an endospore. The endospore is essentially necessary to resist extremes in environment. And so this DNA is a way of protecting the DNA and allowing the bacteria to regenerate after going through a period where it's in a very bad environment. Once a bacteria grow, as I talked about before, they generally form a large group, and a large group of the same bacteria is called a colony.